Alright guys, so what we're going to do today is we're going to pick up where we left off. We're going to basically revisit the charging reaction, just to make sure we understand that. We're going to go through eukaryotic translation, and then we're going to take a look at some of the abnormalities and things like that that can happen either to the DNA or to the chromosomes themselves, and we'll finish out the notes for this particular chapter. So all you really have to do is watch this video today, maybe throw a comment in Google Classroom if there's something you don't understand or you have questions on, and what we'll do is then we'll pick up next week with taking a look at probably like operons and how genes actually kind of turn on and off and how we regulate this whole system of making proteins. Alright, so again, charging reaction. What we got to do is we have to load up our tRNA, our transfer RNA, with the amino acid. So we're going to have our tRNA binding site here in our little enzyme, our amino acyl tRNA synthetase. We're going to have our accepting site. So here comes our tRNA. We've got our side that's going to bind. We've got our anticodon over here. We've got our ATP basically supplying this whole thing with some energy. We have our adenosine monophosphate still hanging out up here. Once we get our tRNA basically docked in this active site, we can then use the power that's left over from our adenosine monophosphate to make the bond right here between this oxygen and down here on this tRNA. So we're going to bond them, our AMP is going to get out of the way, and now we're going to have our charged tRNA with the amino acid. You can see there's the amine end, there's the carboxylic acid end, and there's going to be basically our R group side chain. And now this thing's going to be able to travel over to the ribosome, where we're going to be able to actually then manufacture proteins. So this is where we get into the translation piece. Okay. So what we need to know. Ribosomes, well, they consist of two pieces. It's kind of like a clamping unit. So we've got a large unit and a small unit, and what's going to happen is as the mRNA comes over, it's going to fit in between these two units that are going to clamp down. And that's going to basically allow an area where we can essentially feed this piece of mRNA through. Now, I know you guys might be a little bit old for you, but typewriters used to have ribbons that would feed through. So we're going to feed this thing through there, kind of like a ribbon through a typewriter, and we're going to read all these codons. So we've got 3 different sites, and I know some of you had questions about this. There's the E, the P, and the A site. All right, so the P site. This is the peptidyl, or the growing chain. So right here, this is going to be where the amino acid chain is going to, this is where the amino acids are going to get the polypeptide linkage to link them together and make the actual protein or polypeptide chain. That's going to be coming off of this area right here. The A site, this is where our incoming tRNA that's still charged with the amino acid on it is going to come in right here, and then obviously the exit site. Now I'll probably post another video with this so you can kind of see an animation of how things kind of move through and they leave. Alright, and it'll probably make some more sense. So a combination of me going through this and post another little video, I think you have a pretty good idea of the mechanism of how this actually works. Alright, so here we go. We've got our small subunit. Here comes our mRNA, right? You can see there's our 5' end and our 3' end. Okay, our start part isn't until we get over here. Remember, we've got the caps on there. So we've got our 5' methyl cap, we've got our 3' poly A tail. So we have to read down until we get to that start site. Okay, so what's going to happen? The mRNA is going to bind to the small subunit, and we're going to be looking for methionine. Remember methionine, AUG, there's the anticodon, UAC, and so this is going to basically be our initiation factor. We get an initiation factor here and here, and this is going to be in prokaryotes, right? So these initiation factors are going to basically kind of grab one of these guys. They have an affinity for them, and again, we're dealing with all these 3-dimensional conformations of proteins. They kind of recognize where to go. So these guys are going to align this initiation factor and this initiation factor, are going to align the tRNA and the mRNA so that our large subunit can actually come in here and form our clamp. So once we do that, now notice, when this clamps down, our tRNA, our very first one with our methionine on it, is going to sit right there in that P site. That's where we're going to actually start to grow our chain. So now what's going to happen is we're going to have another one come in over here in the A site. The amino acids are going to have to connect right here, and everything's going to basically be shifting down. Okay? So, here we go. Here's our first one. This is going to be our methionine. Here comes our second amino acid. Now, we've got to form this peptide bond. NCC, NCC. So this has to bind right there. Now, what we have to do is, again, you know what kind of reaction it's going to be. It's going to be a dehydration synthesis. We got to squeeze out the water. So what we're going to do is we got to form that peptide bond in there. As we form that peptide bond, right, this guy here is going to basically move over. They're going to join here, and everything's going to basically be shifting down one step at a time. All right, so it's going to be cyclical. Now this is eukaryotic. It's going to be fairly similar, right? We get an elongation factor. It's going to say, you know, hey, let's start making this process happen. So we're over here. We're going to match the tRNA anticodon with the mRNA codon. That's happening right here, right? So we're going to come in right here. We're going to then move in. There's our elongation factor. That elongation factor is basically guanine diphosphate. It's going to basically get moved out of here, and you're going to see how this chain is going to keep growing, right? Here we're adding amino acids. We're going to add another one on. So we're going to get our peptide bond formation going over here. Now we get the translocation of the ribosome. All this means is that things have to move along, like that typewriter ribbon moving through. So what's going to happen? These two guys have to shift. They have to shift over. This guy's now empty, so it's going to go into the exit site. This guy now has the chain on it. It's going to move into the polypeptide site, and it's going to open up this A site where a new charged amino acid is going to be able to move in and keep this thing going. And it's going to do that until when? Until it gets to a codon that says stop. So everything just keeps shifting along. Like I said, I'll give you a little video to go along with it to kind of walk you through how this works. All right. As far as termination is concerned, we're going to basically end up with a release factor. Now notice the major difference, right? It still kind of fits in here, but it doesn't have quite the same shape, and it's also not carrying an amino acid. So by not carrying that amino acid, it basically causes this whole process to dissociate, and everything's going to leave. Now you'll notice UAA. If you remember from looking at codon tables, that's a stop codon. So the stop codons don't carry an amino acid at all. That's going to be your release factor, and it just causes everything to fall apart because now the cellular machinery, the enzyme machinery can't do its job, so it goes its happy way. All right. Now, this a lot of times will happen right on the rough endoplasmic reticulum. Well, why? Well, it's coated in ribosomes, so it makes it pretty easy. And then what's going to happen is these ribosomes can manufacture the polypeptide and send it right into the endoplasmic reticulum, and it can be moved wherever it needs to go in the cell. So it can begin that basically that whole cell signaling process where maybe something's being moved out or moved through the cell. Okay? So a lot of times we can do that right on the smooth ER, or sorry, the rough ER. All right, so in summary, what are we doing with the gene? We're going to take a genotype and we're going to make a phenotype. We're going to take the actual genes that something has and we're going to express it so you can physically see it. mRNA is made during transcription and it directs protein synthesis and translation. That's kind of the big link, is that mRNA between the DNA and then the final protein product. Transcription and translation can be broken into three parts. All right, essentially initiation, elongation, and termination. You got to start it, you got to move, and you got to stop it. Easy enough. And then obviously eukaryotic gene expression is going to be more complex than prokaryotic for obvious reasons. More genes, more cellular machinery, more things to do and work with. Right? I would suggest, this is a wonderful picture. It's in your textbook. I do not know what page it is. But this literally walks you through the entire process of basically going through and doing transcription and then translation. It's got wonderful little captions here to go along with these pictures to kind of walk you through. It shows you how to make the mRNA, how to cap it and tail it, how to get rid of the introns, how everything's going to come together out in the cytoplasm with the ribosome, how everything's going to move through the EP and A site, and how you're going to build the whole protein. So it's 1, 2, 3, 4, 5, 6, and it walks you through the whole process. I would suggest spend a couple minutes with this. If you can understand this and kind of wrap your head around all the steps that are going on here, you're going to be good to go for understanding both transcription and translation. Now, so far we've been talking about if everything goes right? That doesn't always necessarily happen. If you want to take a look at this table as well, you can, differences between pro and eukaryotic genes, but not everything goes right. Sometimes we get mutations. You can get what's known as a point mutation. That just means one amino acid residue, one letter, is either added or deleted. Now, what does that mean? Well, if I add a letter in, it's going to shift my reading frame. If I take a letter out, that's going to shift my reading frame, and that could potentially cause some serious complications down the road for making our protein, getting the right amino acids, therefore getting the right 3D conformation when the protein goes to quaternary structure. So a single base being added or deleted can have dire consequences. You can also get a base substitution. So let's say we had a G, and then instead of a G, we put a T in instead. Okay? Now, sometimes it makes no difference, right? Remember, the genetic code, there's 64 codons, but there's only 20 amino acids. So it's degenerate. There's more than one way to ask for some of those amino acids. So sometimes changing one base with another one, it's still going to give you the same amino acid, and that would be called a silent base substitution, right? There's no change. You're still going to end up like arginine is what you're normally supposed to have. You change one letter and you still get arginine, okay? There also can be a missense. So now you do a base substitution, but now you're getting a new amino acid. That could be a bad thing, okay? There's also a nonsense mutation. Now, this is where you take a codon, and instead of having it be a normal codon that's coding for an actual amino acid, it's going to get turned into a stop codon. Well, if you have a stop codon, that's going to basically shorten up your protein. So you're going to get what's known as a truncated protein. It's going to be shorter, probably non-functional, or at least less functional. And then, obviously, all of these things here are going to pretty much cause a frame shift mutation or a reading frame mutation. It's going to shift where you're reading, and that shifting of reading is going to affect your end product because of how you have to chunk up the codons. Okay, so up here at the top, this would be an example of just our normal coding and template in RNA, and this is what we should get. And they're giving you examples. Here's a silent mutation. So in this particular instance, it looks like we changed that T to a C, but either way, that still ends up asking for proline. So that would be your silent mutation. I changed one letter, but I still get the same exact amino acid I need. There's no change. So it's a mutation with no effect. Missense. Now, in the missense mutation, you can see what they did over here. It looks like they changed that G to an A, and when they did that, it calls for histidine instead of arginine. So now we've got a new amino acid. So that would be your missense mutation. Is this thing going to fold up the exact same way as this guy? Probably not. So is this one going to be functional? Probably not. And then you get down here to our nonsense mutation, and what they did was they changed the T to an A, and that calls for a stop codon, and as you can see, now we get the shorter or truncated protein, which is probably not going to do its job at all, or at least very well. Now, here's a real-world example of this. You guys, I'm sure, have heard of sickle cell anemia. It results because of one amino acid being changed in the sequence. So normally, you would have glutamine. You'd have GAG is going to be our codon that we need, and that would call for glutamine, and glutamine is polar, but instead we change out that A for a T and we get valine, and valine is hydrophobic. So you had polar stuff, right, that likes water, and now it's hydrophobic, so it's going to try to hide inside, and what that does is it changes these beta subunits of your hemoglobin. Hemoglobin is a quaternary protein made of 4 subunits. There's 2 alphas and there's 2 betas. This is what it would normally be like, and it's nice and round and smooth, which you kind of want that. You want it to be able to grab onto stuff and let go of stuff. As soon as we change this thing and this becomes hydrophobic, now the protein folds up differently, and what's going to happen is our beta subunits change and it becomes sticky. And when these guys become sticky, this hemoglobin, now what that means is that all the hemoglobin that's inside the red blood cell can grab onto each other, and it basically crumples the cell and it sickles it. So that's an example of basically just a single substitution mutation, right? We just put in a different letter, and now we're over here and we got sickle cell anemia. So these simple mutations can happen, and they can have some serious effects. Now, the other thing that can happen is we can also get chromosomal mutations. Now, this could maybe happen during meiosis, obviously, but we can get deletions. So up here in this top example, you can see they've kind of chunked this out into little sections, but here they're getting rid of B, C, and D. So you're missing a whole segment of a particular chromosome. We know that having one more chromosome or one less chromosome can be probably bad news in most cases. Having just a little chunk missing or taken out, also going to be bad news. Duplication. Here we're adding extra information in. So they took the B, C, D, and they doubled it. So now I've got double the information for these particular genes. That's probably going to cause some problems as well. Inversion. B, C, and D got flipped. So now they're going to be read out of order. Could that be problematic? It could be, especially if all of these genes, one basically is tied to the next, and they're all interconnected in some way, shape, or form. So that can be problematic. And then the last one is reciprocal translocation. So this is when two chromosomes swap information. So here you can see the B and the C from this chromosome get switched out for the L and the M from this chromosome, resulting in, or sorry, the K, L, and the M, and the A, B, and the C get switched out, and you get two brand new chromosomes that are probably not going to have the right information and function the way they're supposed to, which means down the road bad proteins and probably some issues. And that's all I got for you for the day, guys. So do me a favor, watch this some point over the weekend. If you have any questions, post them in the Google Classroom comments, and that way we can address it next week. I may also do like a little open tutoring session later today just through Google Meets. I'll post that probably sometime in the afternoon. It's optional, you don't have to show up, but if you want to come in, check it out, have questions or something like that, feel free. All right, talk to you guys next week if I don't see you later today. Bye.